Our next speaker today is Isabella Yepes, and she is a senior in high school, be impressed, in Bedford, New York, and she just told me she was just accepted to Columbia University for environmental engineering. So, welcome me and welcome her.
Good afternoon, everyone. Thank you for the welcome. As she mentioned, I am a senior in high school, and I spent the last 3 years in the wildlife technician program at the Mianus Gorge. It was a 3-year program where I could work with a mentor and take an assessment of the health conditions for 39 wetlands in the Mianus Watershed. So I'll get right into it. Let's see. So on this map, do you guys know what on this map contributes to drinking water for 130,000 people? Well, it's the second marker right there, the Mianus River. It actually has the river provides drinking water to 130,000 people in southern New York and Connecticut. And what's very critical about this river is the 39 wetlands that go along the stream. They're actively filtering the water with their rich vegetation. But there was a problem. As of 2016, there's no information on the individual wetland health for these wetlands. And without this information, local preserves like the Mianus River Gorge cannot fully protect the wetland in the watershed. And so they could be under threat from invasive species, pollution, human land use, etc. And so to solve that problem, I performed the assessment. And I'm hoping that this will help provide the information for to in the future restore the wetlands and improve the whole drinking water. So the 3 main ways I assessed the wetlands health was taking data with GIS. I used Archives for Connecticut and New York. Also through direct observation at the wetland, I looked at the density of vegetation and the hydrology and topography at the wetland. Also, I used a basic way to test the water chemistry, such as just using a test strip and collecting pH, nitrate, nitrite, hardness, chlorine. This data was were inputted into the computer model. I used the MRG's 1998 procedure, and this is a rapid assessment which allows us to assess the overall watershed. So essentially the MRG's procedure generates a score from 0 to 1 for each wetland's ability to perform 8 functions. These are the functions you see here. So I was concerned with how well the wetland discharged groundwater into its surrounding, and how well the vegetation was, and also if it had a good ability to store water. So for example, if a wetland had no vegetation, then it would score a 0, or if it had a very abundant and diverse vegetation, it would score 1. So just show you some results. Again, this was for each of the 8 functions, but this is just one of the results. You can see the areas in dark pink show that those wetlands had high abilities to store water, and that would be helpful in case of a storm or flood, because you know that those are the ones that are kind of safeguarding and providing that buffer zone. This is another map showing the ability for the wetland to discharge groundwater on the left. So you can see the wetlands in dark color are discharging more of the water, and that's what's really kind of the headwaters of the river. That's what's fueling the river's water. And you can see on the right the recharge ability. So this is absorbing the water into the wetland and possibly going into the aquifer and providing local communities with drinking water. So you can see that you might notice that there's kind of an overlap, and that's because there was a strong correlation. So that means it was a 0.94 negative correlation. So that means a wetland either discharged water or recharged water, which is a little expected, but it's kind of interesting result. Just an example of what the procedure generates. So here we see some water chemistry results. Overall, they were in good conditions, but there was a few things that kind of caught our attention. You can see on the top is the chlorine results. Some of the wetlands had not 0 chlorine levels, but then again, it was still parts per million, so it's a small amount. So it's, but just kind of understanding which wetlands are in those conditions can allow us to kind of restore them in the future. So you can see some of them are actually repeated. So a lot of the wetlands that were had higher chlorine levels also had high basic pH levels, and they also had similar results for nitrate levels. So I essentially wanted to quantify like which wetlands were repeatedly in worse conditions. So I kind of gave them a value if they had a range that was out of the ideal. And that's the list on the left kind of shows the wetlands that had the most need for restoration. And then I also wanted to do that with preservation. So this is more of like the wetlands that were in best condition, the ones that had the highest ability to store water, the ones that had the highest ability to modify water quality and clean and filter water. So those are the wetlands on the right. So this is important because before there was like no information on each wetland, so this is kind of giving us a baseline view of the overall watershed. For the ones that were in worse condition, I would recommend eliminating and identify the polluting sources. Also involve the community by outreach and other events. For protecting the wetlands in the best condition, I would recommend fencing in and protecting the notable species, and also using the data to build community interest. So this is just an idea, like as far as education goes, maybe we can have a sign in the wetland, like while you're on a nature trail and you're passing through a wetland, you see the sign that says, did you know this wetland is in the top 20% for the ability to store water in case of a flood? And then that will like get the people who are walking through like more interested and more aware that yes, this is actually like having a value to protect me from erosion or flooding. So just some thoughts. And then also I looked into invasive species. So my method for collecting invasive species data was a little basic. I just counted for how many species were in each wetland. Again, I covered the wetlands perimeter, so this was a rough estimate. But you can see here there's no clear pattern of the invasive species presence in relation to vegetation abundance or wildlife suitability. So here you can see a graph showing the 4 columns. So the column on the left is 0% cover of invasives, and the column on the right is 100% cover of invasives. And you can see the blue indicates the wetlands that had no protection from a local preserve, and those actually seem to be the ones that had the highest cover of invasive species. So this kind of shows that unprotected wetlands do have higher invasive species cover, which shows that the fact that it is protected is helping them with less invasive species cover. So overall, just some conclusions for the overall Mianus River watershed. All the wetlands had above 0.5 for vegetation and wildlife habitat potential, so that shows that they're in a good condition. But then I wanted to see like which ones were in the best condition, and so that would be if they had a score above 0.75, and 82% had ideal health for vegetation, 44% had a high ability to clean and filter water, and then 31% had ideal wildlife habitat potential. So this is again a score above 0.75 after putting the data into the computer model. Just covering again, just conclusions. Unprotected wetlands had higher invasive species cover, which is kind of expected because protected wetlands would probably be more controlled with the invasives. There's also not really a clear pattern of invasive species presence versus the vegetation abundance. And I think this also has to do with the way I measured vegetation abundance. I didn't really control for like the invasive species, so again, this is why there's not really a clear pattern present. But finally, the wetlands with the full protection had slightly higher restoration need than unprotected wetlands, which was a little bit surprising for us, but I think that kind of highlights the importance of doing this study and kind of seeing which wetlands are in the worst conditions so that we can restore them in the future. Let's see, another just kind of like overall picture of like what the project went out to do. It successfully assessed the current health and function for the 39 wetlands. It determined the wetland structure, like the ones that you saw about storage and all the other functions. And also I helped identify which are in the worst condition and most need restoration, and which are in the best condition and most need preservation. In the future, I'd like to see the wetlands identified to be restored, and also about Maggie's procedure. It was a rapid procedure, so I felt like it needed a little bit more like in-depth look. So maybe going more with the wildlife diversity, having more quantitative measurements, because there wasn't like a specific species monitoring. It was more a general overview of is there plant food, animal food present in the wetland. It was just looking at the plants and using that as an indicator for the wildlife habitat potential instead of actually monitoring the specific species. Finally, I think the invasive species effects are complex because the presence of several invasives did not really impact the wetland water quality or structural health, because even though the invasive species are, of course, like as previous studies show, like affecting the biodiversity and vegetation health, the actual water quality and structural health is not negatively impacted as far as our measurements showed by the invasive species, because they're still functioning as plants and filtering the water naturally. So it's just a important note. And then I'd like to thank my research mentor and also the grants. So thank you. Oh, this was just another slide. That's it. Thanks. Yes.
First of all, it's a really interesting topic and you did a great job presenting.
Thank you.
I'm curious, were all of the wetlands, all 39 wetlands, the same type of wetland?
Great question. As far as the
Like forested versus shrub swamps and that kind of thing?
yeah, they were actually different wetland types. So the way the procedure actually accounted for the type of wetland it was. So if it was I think it classified. I'm not sure if it was. It definitely went over like if it was riverine, slope, like on a hill, and depressional if it was like in the basin or valley, because that would actually change how hydrology goes in the river based on like the topography of the wetland. So it definitely did we did account for that in the study, and not all wetlands were the same type. So, but yes, thank you. Yes.
And how much did they pay you?
Yeah, it was for fun.
So question, so you were able to access all the wetlands, that was not a problem?
Actually, there was there's 39 assessed, and there was a total of 41 that we wanted to assess, but 2 of them were inaccessible. One of them was in a private property, and they didn't really allow us to access it, and so we did encounter an issue there, but we got almost all of them done. Except
That's great. And were you able to go back to them more than once?
Yeah, because of the changing seasons, we wanted to make sure we took a kind of an idea that wouldn't be different in the summer versus the winter. So I think we visited twice for each wetland, although with some of them there was no water at the wetland, so we had to we weren't able to take pH samples at some of them.
Okay, great.
Yes.
So when you said that some of the areas were in need of restoration, could you figure out in any of those wetlands what needed to be done as a future engineer? That's kind of thing to think about.
Yeah, I would imagine because some of them I mentioned with the water chemistry results, some of them had I guess dangerous levels for that we expected for chlorine and nitrates. So I would say that might be a pollutant source involved at that wetland that's causing the restoration need. But again, I guess that might be specific to each wetland, and just knowing which ones are in bad condition, we can look further to see what's the actual cause. Yes.
Just how did you measure the correlation between the 2 factors that you were measuring? Things like -0.9.
Yeah,
was it
yeah.
R-squared value?
Yeah, it was the Pearson correlation coefficient. I'm not really sure the exact thing, but I just used Excel. So. Any other questions or? Thank you guys.